So in Chapter 4, Part 2, we're going to be focusing on a lot of those internal organelles, focusing particularly on the nucleus and something called the endomembrane system. So the genetic instructions in a eukaryotic cell, in other words, the DNA, are kept in the nucleus. We say they're housed in the nucleus. And the instructions are actually carried out by the ribosome. So you can think about the ribosomes as the workers in the plant. And the nucleus is kind of like the director or the boss. So let's take a quick look at sorry, just backing up a quick second here for you. The nucleus carries most of the DNA in a eukaryotic cell, but not all of it. Some DNA is found in some other structures. I'll give you a heads up. The mitochondria and the chloroplasts also have DNA. But again, for the most part, the vast majority of the instructions in a eukaryotic cell are going to be found in the nucleus. Now, the nucleus has certain parts to it. There's the nuclear envelope, there's the nucleolus, and then there's the chromatin. So let's take a quick look at all of that. I know the picture is a little bit smaller here, and I'm going to be flopping back and forth because I have the direction, I have some nice information on this slide, but I think the picture is better on the other slide. So let's see what happens here. The first is the nucleus, and the nucleus is probably the biggest, definitely the most conspicuous organelle in the cell. And we say it's conspicuous because it's also something that picks up a lot of dye, and so it's easy to see under a microscope. The nucleus is surrounded by a membrane called the nuclear envelope, and the nuclear membrane that surrounds it is actually two membranes together. So it's a little hard to see in this image too, but there are two envelopes, two membranes that surround the nucleus. Each of those membranes is consistent of two lipid bilayers. So phospholipid bilayer in the inner membrane, and a phospholipid bilayer in the outer membrane. Then there are these holes in the nucleus, and they're holes that are called pores. They're pretty big. They allow things to go into the nucleus and out of the nucleus. And a lot of things that are going in and out of the nucleus are pretty big molecules, like ribosomes and so forth. Ribosomes are going to be made in the region of the nucleus called the nucleolus. And the nucleolus is right there in the center of our nucleus. The nucleolus makes ribosomes, and ribosomes make proteins. Sometimes there's more than one nucleolus in a cell, which is kind of cool. If we look a little bit more carefully at the nucleus, we can say that's where the DNA is found, right? And the DNA itself is organized into these structures called chromosomes. Now, we tend to think about chromosomes as kind of having this X shape, but we'll learn a little bit later in the semester that X shape is really not the way that they look most of the time. It's just the way they look when we can see them under a light microscope. But most of the time, it's like a long string of DNA that's surrounding, like kind of winding around these proteins called histones. And again, we'll learn more about that later. So each chromosome is one long DNA molecule wrapped around proteins. When the DNA and the proteins all come together, we call that structure chromatin. Again, there's a lot of chroma words when it comes to the nucleus. So chromatin is DNA wrapped around proteins, and chromosomes are each kind of long or compacted bit of DNA and proteins. So it's a little confusing right now. It'll make more sense when we get into cell division. the chromatin does condense, and condense is just a word for like coils up. Look how it starts to coil up here, but then is really, really, really tightly coiled up here. So the chromatin condenses or coils up to form these really very clearly separated packages called chromosomes. Again, it's in the nucleolus that the ribosomes are made, for the most part. The ribosomes are made of something called ribosomal or rRNA, and again, we'll spend more time on that when we get to it in another chapter. This is kind of like some basic background for you. Ribosomes are the protein factories of the cell. They're absolutely the place where proteins are made. And ribosomes are made up of ribosomal RNA, also known as rRNA, and proteins. There's two places where you can find ribosomes after they're made, right? Like remember, the ribosomes are made in the nucleolus, which is found in the nucleus. But after that's made, it's shuttled out of the nucleus through those nuclear pores and into the cytoplasm. Once it's in the cytoplasm, it can be found in two locations, either as free ribosomes just hanging out in the cytosol, or ribosomes that are bound to the nuclear envelope or to the endoplasmic reticulum. So if they're attached to a membrane, we say they're bound ribosomes, and if they're just free floating, we call them free ribosomes. So the next thing that we're going to talk about is the endomembrane system. And this is a pretty complicated system inside the cells of interconnected organelles, different organelles all acting together to have your cells function as they need to function. There are several components of the endomembrane system. I'm just going to list them here for you right now, and we'll go into details on them as we go along. So the first is the nuclear envelope. Remember, the nuclear envelope surrounds the DNA, but it's also considered to be part of the endomembrane system. We also have the endoplasmic reticulum, and there's a couple different kinds of endoplasmic reticulum. Then there's something called the Golgi Apparatus. We have lysosomes, vacuoles, and finally the plasma membrane. I say finally, but that's not quite true, because the way that things kind of are passed along from one part of the endomembrane system to the other is through these structures called vesicles. So if we take a look over here, got our nuclear envelope, we have our endoplasmic reticulum, there's rough endoplasmic reticulum and smooth endoplasmic reticulum. Those reticula form vesicles, which you can see right here, and those vesicles carry information to the Golgi. The Golgi then bud off more vesicles, which can do a number of things, including become lysosomes. They can become vacuoles. They can fuse together with other vesicles, or they can fuse with the plasma membrane and allow stuff to leave our cells in very large numbers. So let's focus here first on the endoplasmic reticulum. And the main job of the endoplasmic reticulum is to make stuff. Make stuff. Another way to say that is we biosynthesize. And so we can say that the endoplasmic reticulum is a biosynthetic factory, making stuff, making biological molecules. The endoplasmic reticulum is frequently abbreviated as ER, and the endoplasmic reticulum accounts for like more than half of all the membrane inside of eukaryotic cells. There's just a lot of endoplasmic reticulum. And you notice how it's all folded back and forth on itself. It's also connected directly to the nuclear envelope. So it's communicating with everything that's going on inside the nucleus. I said before, there's two distinct regions of endoplasmic reticulum. There's the smooth ER. It's smooth because it lacks ribosomes. And the rough ER, and it's rough because it's got ribosomes kind of studded all over there. So the rough endoplasmic reticulum, we say, is studded or covered in Ribosomes. You know what ribosomes do. I bet you can guess what the rough endoplasmic reticulum does. Let's talk about the smooth endoplasmic reticulum first, the smooth ER. It has several functions, including synthesizing lipids, making fats, all kinds of fats. In the previous chapter, we learned about 3 categories of fats, including phospholipids and triacylglycerols and cholesterol. So synthesizing lipids. It also metabolizes carbohydrates. That means that it breaks down carbohydrates and it makes carbohydrates. The third thing it does is detoxifies drugs and poisons. So if you get some mild poisoning, that's when your smooth ER comes in and kind of makes those poisons not poisonous. Finally, it stores calcium ions, and that would be very important in lots of functions in your body, including your muscles working and building of bones. The rough endoplasmic reticulum has those ribosomes, right? And those bound ribosomes make and send out, or the word here is secrete, things called glycoproteins. And glycoproteins are any protein that is covalently bonded to a carbohydrate. That root word glyco there, that indicates that you're talking about some sort of sugar. And a lot of these are very long chain polysaccharides. The rough ER makes the proteins, again, specifically the glycoproteins. That's very, very common. And then it puts those glycoproteins into transport vesicles. And those proteins are now surrounded by a membrane. And remember that the membrane is important in almost everything in the cell. Going back here, the rough ER basically is important in making important parts of the membrane for the cell. Now, so is the smooth ER. Remember, the smooth ER makes phospholipids, right? It makes cholesterol. So both the rough ER and the smooth ER help make the plasma membrane. The Golgi apparatus is also membrane-bound organelle that's a series of flat sacs, and those sacs are called cisternae, cisternae, cisternae. The function of the Golgi is to take products from the endoplasmic reticula, both the smooth and the rough, and modify them, make them so that they're working how they're supposed to work. They also do manufacture some macromolecules, so it's part of that making of stuff. And then it also sorts stuff that's gotten from the ER and packages it into transport vesicles and sends it on its way. A lot of people say that the Golgi apparatus is kind of like the United States Postal Service, shipping and receiving. Lysosomes are made and are part of the system, the endomembrane system, because they are a form of a vesicle that's made at the Golgi apparatus. And lysosomes have a very specific role that's similar to the digestive system in your body, the stomach. It breaks down bigger molecules into smaller molecules. So we learned the word hydrolysis in a previous chapter, and we learned the word enzymes in a previous chapter. The lysosomes are filled with hydrolytic enzymes. Again, these are enzymes that break down or digest larger molecules. Lysosomes work very well in an acidic environment, kind of like how your stomach is acidic. So lysosomes really do fit this idea of digestive compartments. The shape of the proteins protects them from being digested by the enzymes. So we've got some protection going on there based on shape. Remember, we talked about protein shape the tertiary structure and the quaternary structure in the last chapter. Not only do lysosomes act as a digestive system in general for any important food products that come in, but we can also think about how they do that. And some cells will actually engulf food in a process called phagocytosis. That literally means cell eating. So it's eating, it brings in food in this form of something called a food vacuole. All right, so here's some phagocytosis, and it's bringing in a bunch of food, and that food is inside this vacuole. The lysosome merges with the food vacuole, and the digestion happens inside that merged structure. Lysosomes also help recycle any organelles that aren't working properly inside of the cell. For example, if a mitochondria breaks down and stops working, then a lysosome will come along and it will digest that mitochondria and kind of recycle or reuse some of those structures and build other things. Now, I mentioned food vacuoles in the previous slide, but vacuoles in general are very diverse compartments. This is kind of like a generic term for large vesicles that are made by the endoplasmic reticulum, the Golgi apparatus, or as we saw in the previous slide, if it's a pinching off of the plasma membrane. Whatever is kind of packaged inside that vacuole is what the vacuole does. So vacuoles can have different roles depending on the stuff that's inside of it. And remember that each of these membrane-bound organelles, they each have their own membrane, which tells what goes in and out. So the stuff inside a vacuole is going to be different from what's inside the cytosol. I just mentioned before the idea of the food vacuole, and the food vacuole is formed by this process of phagocytosis. I did grab a picture there. You can see it at the top of a food particle being engulfed by a plasma membrane, and that engulfing is called phagocytosis. Then along come the lysosomes, and they surround that food vacuole, and then everything's digested. You can also have something called contractile vacuoles. Contractile vacuoles are very cool structures. You can see them actively working in things like paramecium. They pump water, excess water, outside of the cells, and basically prevent those cells from exploding because there's too much water coming in. So contractile vacuoles contract or compress, squirting water outside the cell to get rid of it so that the cell doesn't explode. Some vacuoles that you find in plants and fungi have structures that are like lysosomes, but have different compounds, so we don't call them lysosomes. So we give them other names, but they're very much like lysosomes. And then, of course, we have the big central vacuole inside of plant cells. Right here, central vacuole can be one of the largest structures that you see inside of a plant cell, especially if that plant cell has just been watered or something along those lines. Central vacuoles hold inorganic ions like potassium and chloride, as well as lots of other things like pigments. Flowers have different colors, and the colors are held, the pigments that make those colors are held inside the central vacuole. So just a really quick review of the endomembrane system, because it is a pretty complicated system. Information inside the nucleus, which is defined by the nuclear envelope, is read and transmitted through the nuclear pores out into the endoplasmic reticulum. Specifically, we're going to have a lot of that information going to the rough ER, because that's where you find those ribosomes. The rough ER and the smooth ER are both continuous with the nucleus. information that is manufactured in the ER are butted off from the ER into structures called vesicles. Those vesicles move towards the Golgi apparatus and are absorbed by the Golgi apparatus. As they move through the Golgi, they are modified and changed and packaged into different structures. Some of those structures can stay inside the cell and are lysosomes. Other transport vesicles carry proteins to the plasma membrane, and those proteins are secreted or released to the outside of the cell. And that is everything about the nucleus and the endomembrane system. We're going to move on to Chapter 4, Part 3 in just a minute.